A very good morning, everyone. I'm Shruti of grade 11A. Today, on the occasion of Science Day, I'll be presenting a seminar on the topic The Intersection of Biotechnology and Medicine. The medical field has evolved phenomenally that even lethal diseases such as tumors and cancers and autoimmune conditions can be treated and addressed with advanced and time efficient techniques. In the past, a disease like cancer meant death. But now, survival rates have improved and patients are getting into remission faster. How has this been achieved? How are new drugs invented? How has the medical field even advanced? How did epidemics slowly vanish? All these questions can be answered if we dig back to the past. Scientists always believed that the genome could be altered and manipulated. We have 25,000 coding genes in the human body, and mutations in any of these could cause fatal abnormalities as they disrupt cellular machinery. When the knowledge of the central dogma became clearer, researchers began harnessing methods to alter genes in a quest to improve the quality of human life. And that was when biotechnology was born and the term was coined in 1919. But what is biotechnology? Biotechnology is the integration of natural sciences and engineering in order to achieve the application of organism cells, parts, and their molecular analogues for products and services. This concept encompasses a wide range of procedures for modifying living organisms according to human purposes. It includes artificial selection and hybridization through breeding programs. Modern usage also includes genetic engineering as well as cell and tissue culture technologies. But how is biotechnology related to medicine? Bioengineering and biomedical engineering are the two subsets of biotechnology that are closely related to medicine. Bioengineering is the application of the principles of engineering and natural sciences to tissues, cells, and molecules. Relatedly, biomedical engineering is an overlapping field that draws upon and applies biotechnology, especially in certain subfields of biomedical or chemical engineering, tissue engineering, biopharmaceuticals, and genetic engineering. Here's a brief overview of how biomedical engineering can be applicable in the medical field. Tissue engineering is used clinically to study diseases under in vitro conditions. Genetic screening like COVID RT-PCR being the best example as well as relatable. And then we have hormone manufacturing and synthetic protein manufacturing for diseases like diabetes mellitus type 2. Prenatal testing of genetic diseases like Down syndrome and inherited conditions can fall under this category. Often there is no cure for genetic diseases since they cause faulty functional changes in our genetic code. But gene therapy allows doctors to inject modified new functional genes that have been engineered in the lab using vectors such as viruses and bacteria that are modified into the human cells or specific targets. Biomedical informatics and AI are used for medical teaching and digitization of patient records and data. In this presentation, we will focus on nanotechnology and nanomedicine, CRISPR-Cas9, and genetic engineering's applications in the medical field. Let's begin with nanotechnology. Nanotechnology is defined by the Nanotechnology Initiative as the manipulation of matter with at least one dimension sized from 1 to 100 nanometers. The medical application of nanotechnology is called nanomedicine. What are the benefits and advantages of nanomedicine? Nanotechnology has been applied to increase absorbability of a certain drug. Nanoparticles are used in order to serve this purpose. Nanomedicine is also applicable for drugs that are absorbed too quickly and are removed from the body as waste before treatment can even be effective. The accumulation effect is produced in certain inflammatory diseases like rheumatoid arthritis because blood vessel leakage is a common characteristic of inflamed tissue. Nanoparticles provides site-specific drug delivery for treatment of inflammatory diseases through the same accumulation effect as even found in solid tumors. So it's site-specific, and molecular targeting by nano-engineered devices could potentially prevent invasive surgeries. It also serves as an easier disease detection mechanism with lesser side effects. The medical field has progressed from the concept of therapies like the one-size-fits-all mechanism into personalized molecular targeted therapies to improve patient care, and it is suited for the body and microenvironment of the specific patient. But what's the principle behind nanotechnology? How does it work? Well, nanotechnology works by injecting small nano machines into your body, which are pre-programmed to navigate themselves to the site of the disease and function to repair tissues or clear your arterial lumen because of the accumulation of plaque, and even attack cancer cells. Here's an infographic of how orthopedicians could use nanomedicine. As you can see, for bone cancer, they could use nanomedicine for delivering the chemotherapy drugs. They could use it for bone tissue engineering, especially in excessive demineralization of the bones due to certain conditions. It could be used to implant nanostructured materials, and it could also cause surface modifications and coatings, and it could also be used to create new osteoblast cells. And here's another infographic of how it could be applied to treatment in cancer. It could be applied to cancer by enhancing immune response by releasing certain CTLs called cytotoxic T lymphocytes, or by reducing the immune toxicity. Summing up, nanotherapeutics has 6 major applications. As dystopian as it may sound, nanoparticles like nanolipids are prospected to be replaced by nanorobots or nanobots in future, as you may have seen in the movie called Inner Space. So here are the 6 applications as you can see on the slide. Drug delivery, targeted therapies, gene delivery through nanoparticles and nanolipids, detection and diagnosis, molecular imaging, and biomarker mapping. Our next technology of focus is CRISPR-Cas9. CRISPR-Cas9 is an acronym for Clustered Regularly Interspaced Short Palindromic Repeats. A mouthful of a word, isn't it? So let's stick with CRISPR. And Cas9 is an endonuclease. This system acts as molecular scissors to cleave specific regions of DNA. CRISPR is a family of DNA sequences found in the genomes of prokaryotic organisms such as archaea and bacteria. These sequences are derived from DNA fragments of bacteriophages that had previously infected the prokaryote. They are used to detect and destroy DNA fragments from bacteriophages in certain subsequent infections. Hence, these sequences play a crucial role in antiviral defense system of prokaryotes and provide a form of adaptive immunity. CRISPR are found in approximately 50% of the sequenced bacterial genomes and nearly 90% of the sequenced archaea. It is a gene editing technology that has been causing a major upheaval in biomedical research. It makes it possible to correct errors in the genome and turn on or turn off genes in cells and organisms quickly, cheaply, and with relative ease. Cas9 is an enzyme that uses CRISPR sequences as a guide to recognize and cleave specific strands of DNA that are complementary to the CRISPR sequence. Cas9 enzymes together with CRISPR sequences form the basis of the technology known as CRISPR-Cas9 that can be used to edit genes within organisms. This editing process has a wide variety of applications, including basic biological research, translational research, and even in clinical trials as well. Suppose AGGUCCA is the CRISPR sequence, Cas9 would cleave UCCAGU in the genome, where A, G, C, and U are adenine, guanine, cytosine, and uracil base pairs or nucleotides, which are the monomers of DNA. The discovery of clustered DNA repeats took place independently in three parts of the world. The first description was called CRISPR from Osaka University in the year 1987. They accidentally cloned a part of a CRISPR sequence together with the IAP gene in the E. coli organism, which was their target. The organization of the repeats was quite unusual, because it wasn't studied before, and these repeated sequences are typically arranged consecutively without interspaced or different sequences. They did not know the function of these interrupted clustered repeats back then. However, in the year 2005, three independent research groups showed that some CRISPR spacers are derived from the phage or extrachromosomal DNA, such as plasmids. In effect, the spacers are fragments of DNA gathered from viruses that had previously tried to attack the cell. The source of the spacers was the sign that CRISPR-Cas system could have a role in the adaptive immunity in bacteria. Researchers then studied a simpler CRISPR-Cas9 system from the Streptococcus pyogenes that relies on the protein Cas9. The Cas9 endonuclease is a four-component system that includes two molecules, CRISPR RNA and targeted activating CRISPR RNA, also called tracker RNA. Jennifer Doudna and Emmanuel Charpentier re-engineered the Cas9 endonuclease into a more manageable two-component system by fusing the two RNA molecules into a single guide RNA, also called sgRNA, that when combined with Cas9 could cleave and cut the DNA targeted sequences specified by the guide RNA or the gRNA. This contribution was so significant that it was recognized by the Nobel Prize in Chemistry in 2020. By manipulating the nucleotide sequence of the guide RNA, the artificial Cas9 system could be programmed to target any DNA sequence for the cleavage. Here's a brief overview of how CRISPR-Cas9 works. As you can see in the diagram here, the genomic DNA is the one that belongs to your human body. And then you have the PAM sequence, or the protospacer adjacent motif, which is adjacent to the CRISPR sequence we are trying to target. The CRISPR-Cas9 system consists of two major molecules that incorporate a modification into the DNA. An enzyme acts as a pair of molecular scissors that can be cut two strands of DNA on a specific area so that removal or addition of DNA can be performed. The second is a slice of RNA or protein called RNA guide or guide RNA, and this is a small piece of about 20 base pairs long of pre-designed RNA sequence placed into a longer RNA scaffold. This scaffold is not our focus here, the guide RNA is. This scaffold section binds to the right part of the genome, that is the CRISPR part, and it can be used to form the gRNA DNA complex, and this binding is specific. This means that the guide RNA can only bind to the targeted sequence and no other genome regions. And then in the DNA sequence, the Cas9 follows the guide RNA to the same position and makes a cut through both the strands of the DNA. The cell at this point understands that the DNA is damaged, and as you can see, a donor DNA here, the donor DNA acts as a template to form the complementary DNA to repair the cut. Researchers can add a putative template or a putative target containing changes altering the existing sequence in the genome, and this is exactly how we can correct a disease-causing mutation in the human body. Here's a brief comparison of CRISPR with other technologies like the zinc finger nucleases and TALEN, and why it's superior. The major reason is that it is an all-in-one gRNA Cas9 vector system, and it has a multi-gene editing possibility, which is extremely feasible as well. And here's another animation of how the CRISPR-Cas9 system works. But our focus is CRISPR-Cas9's applications. It could be used in cell and animal models, like they created an organism called a transparent frog using the CRISPR-Cas9 systems, and it's extremely fascinating to read more about. Human disease therapies, new drug screenings. It could also be used in agriculture. It could be used in embryonic and gene editing in an in vitro environment so as to prevent any diseases like Down syndrome and other inherited conditions. And it could also be used in detection and diagnosis of nucleic acids. In July 2019, CRISPR was performed experimentally to treat a patient with sickle cell anemia on a 34-year-old woman. In February 2020, progress was made on HIV treatments with 60 to 80% of the integrated viral DNA being removed in mice and some being completely free of this HIV virus. In March 2020, CRISPR modified virus was injected into a patient's eye in an attempt to treat Leber congenital amaurosis. In future, CRISPR gene editing could potentially be used to create new species or revive extinct species from closely related ones. This was the broad aspect of technology CRISPR-Cas9 that we could apply this into the medical field as well as other parts of biology, but this is actually the technical aspect of its application, including transgene free modifications, gene expression regulation, viral resistance, chromosome and its structure and the number manipulations, as well as site specific DNA integration. Scientists have curated targets based on the donor DNA template for certain diseases. Based on my studies along with my professor about acute myeloid leukemia, glioblastoma, and triple negative breast cancer, we found the following putative targets for the CRISPR-Cas9 technology which we could apply to treat the following three cancers we are studying. And the ones that are highlighted in yellow are actually more established in the field, whereas the unhighlighted ones were after a lot of research and literature review we did. And finally, we have genetic engineering. Genetic engineering is the direct manipulation of an organism's genetic material. Modifications span from single nucleotide switches to exogenous gene expression to produce the desired characteristic. It has been promising for its precision, accuracy, and potential to treat lethal diseases. It is a set of technologies used to change the genetic makeup of cells, including the transfer of genes within and across species boundaries to produce improved or novel organisms. New DNA is either the recombinant DNA or it is unartificially synthesized DNA. It could also be used to even knock out certain genes apart from editing or inserting certain genes. The new DNA can be either inserted randomly or at a specific target site of the genome to improve precision. Here are the applications of genetic engineering. Genetic engineering is extremely versatile and it has so many applications in many aspects of biology, but since the focus is medicine, here are the following applications. It has many applications to medicine that include the manufacturing of drugs, creation of animal models that mimic human conditions to study diseases, and even perform gene therapy. One of the earliest uses of genetic engineering was to mass produce human insulin in bacteria, and now this application has been applied to human growth hormones, follicle stimulating hormones, albumin, monoclonal antibodies, anti-hemophilic factors, vaccines, and many other drugs. Genetic engineering is also used to create animal models of certain human diseases. Genetically modified mice are most commonly genetically engineered animal models, and they have been used to study and model cancer, and it's called the oncomouse, and it is also used to mimic conditions of obesity, heart disease, diabetes, and arthritis, substance abuse, anxiety, aging, and Parkinson's disease. Potential cures can be tested against these mouse models before clinical trials. Germline gene therapy would result in any change being inheritable, which raised a concern within the scientific community, and they even called for the stoppage of research based on germline gene therapy because it could potentially be used for enhancement, modification, or alteration of human beings' appearance, adaptability, intelligence, character, and behavior, and this is not ethically right. Researchers are altering the genome of pigs to induce the growth of human organs with the aim of increasing the success of pig to human organ transplantation. Scientists are creating gene drives changing the genomes of mosquitoes to make them immune to malaria, especially of the female Anopheles mosquito and the Aedes aegypti mosquito, and then looking to spread the genetically altered mosquitoes throughout the population in the hopes of eliminating the malarial disease. The biggest topic is genetic engineering and ethics. Deciphering the human genetic code may lead us to prevent or exterminate human diseases bonded to the anomalies in DNA. Yet, it is well known that genetic code could also contain information about the physical aspect of an individual. Playing with genetics could result in people being concerned with producing aesthetically beautiful children rather than being concerned about the health benefits of genetic technologies. The modification of genetics could lead clinical procedures to change the color of the eyes, your skin, your hair, your height, ensure a symmetric face, a symmetric body, and any physical aspect you could imagine. But the ethical issue would primarily consist of using this developing technology for the well being of a person against it to predict and alter how a person would look. Should we play God? And the second is the application of of experiments on human participants. One of the most important ethical aspects to consider when we look at genetic engineering is the application of these experiments on the human body. As most of the drugs and clinical therapies are tested on animals and then on humans in clinical trials, the manipulation of the human gene should be tested in order to examine the results. However, the impact of any alteration of the genetic code could have major consequences on the person as well as their descendants. And most of the times, the participants in these clinical trials are actually people with extremely less rates of survival and terminal illness, and they use this clinical trial as a last hope of their survival. Consequently, they approve any test done hoping for positive result. However, there was one serious genetic accident where the patient passed away 48 hours of the clinical genetic trial because of multiple organ failure and sepsis. And finally, it is exclusion of humans with genetic abnormalities. Once we have full knowledge about genetics, it is said that humans could have a card with all their genetic information. At this point, the possibility of an individual with healthier genetics to discriminate a human with poor genetics would greatly increase. And this is not what is a good thing for a society to progress harmoniously. And genetic engineering must be used for the benefit and betterment of the society and not for aesthetic and other non ethical purposes. Future prospects for the medical field with biotechnology, hopes and challenges. Genetic engineering has developed at a quickening pace in the relatively short period since a successful experiment in the middle 1990s. In the middle 1990s, genetically modified foods were being sold in the supermarkets called the flavor saver tomato, where engineered tomatoes were engineered in such a way that the ethylene production reduces, and thereby the tomato could have a longer shelf life. Now crops are genetically engineered to be able to survive in conditions they wouldn't normally be able to handle. But genetic engineering hasn't and won't stop here. It would even continue to produce hormones and vaccines and other life saving drugs. How we as species solve these problems will not only tell us something about the global landscape of moral decision making, but it will also define precisely where the human race will end up over the next few generations. It is not an exaggeration to say that genetic engineering could totally alter the way we live, and these changes won't necessarily be positive, and that's why we must use science in a mindful aspect. Gene therapy, still a relatively new science, has been able to cure some of the most endemic diseases of the modern age. In 2007, a person was cured of HIV through stem cell transplantation. In 2011, 3 years after his therapy had finished, no traces of HIV were found in his blood. Two patients were also treated for melanoma in 2006 using killer T cells, which were genetically made to attack the cancer cells in the bodies. Fast forwarding to the 2010s, genetic engineering has been undergoing a veritable revolution. Around a decade ago, scientists discovered a new technique in genetic engineering that focused on using the the genes of bacteria to edit the genes of other organisms, and this was what we discussed in our presentation called CRISPR. Some scientists have suggested CRISPR is 4 times as efficient and far easier to use than other gene editing practices and can cut down the process of genetic engineering from months to days. But there's still a long way to realize the complete potential of genetic engineering, but ethics must be followed to improve modern medicine. Science should not be used for destruction, but rather for the advancement of human life. This must be kept in all our minds as future scientists. As Madame Curie once said in her speech in the United States, scientific work must not be considered from the point of view of its direct usefulness, but it must be done for itself, for the beauty of science and the passion of science. And like radium, it might become a benefit for the humanity. Thank you so much for listening to the presentation, and I hope you liked the presentation and learned something about the intersection of biotechnology and medicine.